Good afternoon, class. And we have a practical to do. A practical is about a microscope. We talked about two types of microscope, right? You can remind as one, light microscope, good. Another one, yes, electron microscope.
If you want to slide the stage, you use this knob, this under the stage. This one moves it left and right. This one moves it up and down.
Again, we also talked about the parts of microscope. Unless you know the parts, you can be able to use it, right? So if you look at this microscope, it's the same one that's drawn in your books, okay? So we have very many parts here. So let's start with the part I'm holding. How do you call the part I'm holding? Two parts. Mohammed, how do you call these two parts? Yes, the eyepiece lens. So this is where you're going. Good afternoon, class, and we have a practical to do. A practical is about a microscope. We talked about two types of microscope, right? You can remind as one, light microscope, good. Another one, yes, electron microscope. And we said light microscope uses light to magnify objects, okay? Electron microscope uses electrons to magnify objects. Is that okay up to that point? Good. So today we have a light microscope here, and we want to see the procedure of using a light microscope and how to prepare specimen to be viewed under light microscope. Are we together? Now, before we start anything, we have to know what a microscope is and what it does. What is the function of a microscope? Zakaria?
To make small objects look bigger.
To make small objects look bigger. Another person? Yes, so we say a microscope is used to magnify objects. So objects that appear very tiny can be made to be big under a microscope, and that's what we want to see right now, okay? We talked about animal cell and plant cells. So with our naked eyes, we cannot be able to see the internal cells in organisms, right? But using a microscope, we can be able to see the cell organelles, so that when you talk about a mitochondria, you can be able to see it under a microscope. Is that okay up to that point? Good. And again, we also talked about the parts of microscope. Unless you know the parts, you can be able to use it, right? So if you look at this microscope, it's the same one that's drawn in your books, okay? So we have very many parts here. So let's start with the part I'm holding. How do you call the part I'm holding? You cut up your hand, Akram? The limb. So the limb is the part that the user of the microscope holds as he or she uses the microscope. How do you call this part, Mulki? The base. This is where the microscope rests. So when you put it down, it rests using through the base. How do you call these two parts? These two parts, Mohammed. How do you call these two parts? Yes, the eyepiece lens. So this is where you're going to put your eyes as you view. How do you call this part? Stage. And we said the stage is where the specimen is placed as you view it under microscope. There is these four. How do you call these four? Najat? Objective lens. And remember, we said objective lens, it depends with the view you want. If you want it to appear bigger, big, or the biggest, you use this objective lens. And they are labeled. So each of them, for example, this one is labeled objective lens. And they are labeled. So each of them, for example, this one is labeled. If you see, this one is magnification 10. So this one can only magnify an object up to x10. Are we together? If you look at this one, is 4. Can only magnify up to 4. When you look at this one, is 100, right? This one can be very big. X100 means it magnifies 100 times its normal size. Then we also have this one inside here, 40. Okay? This one here, 40. So objective lenses varies according to how they are used to magnify objects. How do you call these ones? These wheels? Someone has the answer. Yes, Kifa. Coarse adjustment wheels. So what's their function? So this one, if you rotate it, is going to move this the stage, right? It can move it up or down, depends with how you rotate it. So when you rotate like that, you see it's coming up. When you rotate it anti-clockwise, it's moving down. Are we together? So if you put your slide here, if you rotate it like this, it comes closer to the objective lenses. If you rotate it this way, it moves down. Is that okay up to that point? So that's all about the parts. And we have a part here. I don't know whether you can be able to see it. there, right? Are you seeing it? So that's where we have a mirror and the condenser. They are inside there. Okay, so that's where we have the bulb, the mirror and the condenser. Are we together up to that point? So this microscope is a modern one. It has a bulb inside it, okay, to improve the visibility of whatever you are going to view under the microscope. Any question up to there? Good. So that one is all about this device. So we have finished. We are through with this device. Okay. Now I want us to move to the second thing before we start using it. Preparation of specimens to be viewed under microscope. So we talked about some ways or the procedure of preparing a specimen. So anything you want to view under microscope is a specimen. For example, here we have this one. This is an onion, right? So this one, now when we are going to use the microscope to magnify it, we are not going to say it's an onion. We are now going to say it's a specimen. Okay? Are we together? So we can say this specimen Y, and we know it's an onion. Okay? Good. So who can tell me the first way of preparing a specimen? If you have your specimen like this, how do you prepare it? The first step, the very first step is which one? Akram? Staining. Okay, staining is a step, right? Okay, but you cannot stain it when it's like this. Can you stain it when it's like this? The first step, Mulki, thinning, right? So what is thinning? Yes, Zakaria. Yes, you cut it into, not small, but into a very thin. Okay, you make it very thin so that it allows light to pass through it very well, so that as you are viewing, you'll be able to view all the contents of that cell. Are we together? So the first thing, thinning. So you have to ensure that as you cut it in a thin layer. Are we together? What do you use to cut? A sharp objects. It can be a scalpel. We have one here. It can be a scalpel. You can use a razor blade. You can use a very sharp knife. Okay, to cut it into a very thin section. That's one, thinning. Another one, yes, Halima? Staining. Good. Staining is another one. So what is staining? Adding drops of, you cannot use water. This solution here is called iodine solution. Iodine solution. It's not blood. Okay, so this solution is what we are going to use to stain. Or why do we do staining? To make it appear what? Clearer. So if you have thinned, yes, we want to view it. You are going to see some parts, right? Now for you to make the parts more clearer, you are going to stain it. So you are going to add drops of iodine solution. Are we together? And now as you are staining, we have something called, let me show you. These ones here, okay? There are very many. These ones. Who can tell me the name of this one? How do you call these ones? Oh, let me remove one so that you can be able to see. This one? Mohammed? Cover slip? No, but you have tried. Yeah, these are called slides. Are we together? These are the slides. So after you have cut your specimen, you are going to put it on a slide like this. So when you put it here, this is where you are going to stain it from. You cannot stain first is when you put here. You're going to put it here, the thin layer you have cut. Then you stain it by adding drops of iodine solution. We added drops, so you cannot come and start pouring this one. No, we added drops, and we have something called a dropper. So as you're adding drops, you use a dropper to add the drops of iodine solution. Remember, as you're going to stain it, this is liquid, right? It's going to spread in this slide. So what are you going to do? You're going to wipe it, because the moment you wet these lenses, you're going to destroy the lenses, right? And that's not a safety precaution when using a microscope. So you ensure you don't wet the parts. So when you stain it, the liquid is going to spread. So you wipe, or you use a very soft tissue or the serviettes, because those ones are able to absorb the iodine solution. Are we together? That one, you do it before you put it in the microscope. Okay, so you're going to do that in your working table. After you've done that, now we said the third step, use cover slips. So how do you use cover slips? Now cover slips, now where now you put your specimen at the stage. Then now cover slips are these ones. There are very many here, right? So these are the cover slips. You're telling me I've held hair. So these are the cover slips. So after you have stained, you have stained, you use the cover slip. All these ones you do it before you introduce it to your microscope. So after you have done all those three processes, now you bring it to the stage. You bring it to your stage. So in the stage also we have a clip, this one, that is also used to hold now the slide in place. So the slide cannot move because it is held by this loop, the clip. Are we together at that point? Yes. Now that one, if you do all that I've said, we have prepared a specimen to be viewed under a microscope. Are we together? Now your work is now to view. You come to the eyepiece lens. Come to the eyepiece lens. Now you view. It depends now with the, how do you call it, the objective lens you want to use. And also we have eyepiece lenses. They are here. So just rotate the wheel so that you can be able to know which one do you want to use to view your specimen. Are we together up to that point? There is another thing I've learned with this one. So if you look at these slides, they are labeled. They have different labels. So it means they have already made the specimens. So all of them are in this slide. So if, for example, this one is stomach section, this is for animals. So if you want to view a stomach section of an animal, it's already in the slide. So you're not going to look it from an animal, then you bring it. It is here. Okay. So what you're going to do is just to view it. They have made our work very easy. Okay. Are we together up there? So all the slides have the specimen to be viewed. Are we together? Now we are going to do this. We are going to come here under my guidance, all the steps we have talked about. Okay. Then we view so that at least each and every one of you can be able to know what we are talking about. If we talk about magnification of using a microscope, so that at least you can be able to use it.